Hello everyone, this is solving past paper 3-1, October, November 2014. Now in this lab we have a goal which is basically to study the effect of different enzyme concentration on the rate of reaction. Now the enzyme is a lipase enzyme, and as you can see the independent variable here is the different enzyme concentration, and we're going to measure the time needed for the substrate to be hydrolyzed. Now the substrate for lipase, as you can see, is triglyceride, and triglyceride get broken down into fatty acid and glycerol. Now, as a piece of information, in this experiment, basically, we are going to use the color indicator to indicate the end point for this reaction. Now, the end point for this reaction, as it shows in the question paper, is as follows. Now, we have orange and yellow color. The orange one is the original color before the reaction starts, and once it reaches the yellow color, that means the reaction ends up. So basically what you're going to do is to make different concentration of enzyme. And as you know, guys, from previous videos, as well as you know from practical skills, in order to do the different concentrations, simply you need to go through the dilution. Now, as it is stated in the question paper, you need to carry on the simple dilution. You're going to start out of the stock solution, which is 5% of enzyme. What you need to do is to go, or to decide what concentration you go. So I do suggest making 5, 4, 3, 2, and 1%. It's going to be easier to obtain. So simply you need to decide. You need to calculate how much you're going to take from the stock, as well as how much you're going to add of water. And simply you can do that by following the formula C1V1 equals C2V2. And in order to do 10 centimeter cubic, as it is stated in the question asks you to prepare 10 centimeter cubic as V2. In order to do that, simply you need to get 10 centimeter cubic of enzyme and 0 water to get the 5, and then 8 of the enzyme, 2 from the water in order to make the 4. And you can see the rest of the concentration as it shows in the table right here. So after you make your concentration, I'm going to show you how our students made their concentrations. So simply here's the enzyme, which is the stock, and then they made the 5, the 4, the 3, the 2, and the 1. How did they do that? Basically, the first one was done by taking 10 centimeter cubic of the enzyme and placing no water. Okay, that's for the 5. For the 4, they took 8 of the enzyme and then they added water, and that's for the 4. For the 3, they took 6 centimeter cubic of the enzyme and then they added the 4 water, and then they carry on making the 2 and the 1. The 2 by taking 4 of the enzyme and 4 water, and the 1 taking 2 from the enzyme and basically 8 water. And that can be done simply by using the syringes in the that are provided by the examiner or by the lab technician. Now, after you are done with making those different concentration, what you need to do is to find out or to test the effect of different enzyme concentration on the rate of reaction. And in order to do that, simply what you need to do is to get a test tube and to fill 2 centimeter cubic of M, which is milk. Now, milk in this experiment contains the triglyceride, so which is the substrate of the enzyme. You're going to add the indicator, 2 centimeter cubic of the indicator. You're going to add 2 centimeter cubic of solution A, which is an alkaline. And then you're going to add the enzyme. So one test tube would have the 2 centimeter cubic of the enzyme, 5%. Another test tube, 4. Another test tube, 3. And 2, and then 1. And you're going to make a 6th test tube that has the 0 enzyme. And instead of the enzyme, you're going to add water. And that's what is called control experiment. Now, you're going to place your enzymes in the test tubes, as well as the substances I talked about, in order to reach the end point. Now here's the end point. Here is the end point, and here is the original. Here is the original the original color. After you leave it for a specific time that you should measure by stopwatch, you reach this end point, which is yellow color and the original one is the orange color. So this one basically it's a zero concentration, and that's the control, and that's the main purpose of using the control, which is the controlling the results. Now after you do your experiment by placing different concentration in different test tubes and record the time, now here is the results from our students, one of our students. Okay, now 4%, 1%, 2%, 3%. Now guys, as a piece of advice, you need to place the results from the smallest to the largest all the time. The independent variable should be from the smallest number up to the largest number. So here's the result. Now as it shows, as the concentration of enzyme increases, the time needed decreases. Okay, so if you plot your results, you're gonna see that the more the concentration, the less the time needed. Now in this exam actually, or this question paper, you don't need to plot, but I'm telling you, if you are going to plot the results, then it's gonna shows a graph that looks like this. It's gonna decrease up to a specific point. Now after that point, it would level off. So let's assume you use 10% or 20% or etc. The more the concentration, it's gonna take at the same time, because the enzyme is not anymore a limiting factor. Okay, now the red line right here, the red triangle, represent how would you make or calculate the initial rate of reaction. Basically it is a very simple experiment, and it's very easy one. Thank you for watching and have a nice day.